you for the invitation. As mentioned, I'm because of the dual roles that I hold, I'm giving this talk from Geneva, and Walter has kindly agreed to share my slides. I hope the connection will hold. Otherwise, I'll quickly reconnect. So I'm going to present to you today on a project which is a very interdisciplinary grant that I have the pleasure and honor of leading, and the SENZOR project. And SENZOR is a name or is an abbreviation for a socio-ecological network approach to understanding zoonotic, outbreak, and risk. We're at the beginning of the research, so in the first year of the research, and I'll explain that a little bit. And we really, it's a team of interdisciplinary scientists who are trying to generate a completely new understanding for how we can assess and understand zoonotic risk. So I thought that's sort of to answer the first question Walter asked me. It would be interesting for this group of researchers to see really the approach we're taking and how we work together. And I will try and address that a little bit. So in my slides, and if we go to the next slide, I'll introduce the grant. I'll introduce the approach we're taking, the different work packages, and then I'll go a little bit in depth on the field work that we are about to begin in one of the countries, the Gambia, and highlight the anthropological work we do, which for me as a social scientist is the work package that I am most closely involved in. And then I'll offer some reflections of the next 20 minutes or so, and then hopefully we'll have a chance for a bit of discussion. So as I mentioned, and as you can see on this slide, the list of partners involved are listed here. So first of all, it's the Center for International Health Protection at the RKI, Germany's National Public Health Institute, and I'll say a little bit about that center in a moment, just so you know the positionality. Then we're working in two countries, the Gambia and Nigeria, and it's very much a collaborative partnership. It's with the Africa Center for Excellence for Genomics of Infectious Diseases at Redeemer University in Nigeria. And in the Gambia, it's the Medical Research Council unit there, which actually now it falls within the remit of London School of Hygiene. So it's sort of quite nicely combining my two hats. We also work with the French National Institute for Sustainable Development, IRD, where our anthropological colleague is based, as well as the Natural History Museum in London. We were awarded at the end of 2022. We started last as a 4-year grant, and it will run until 2027. If we go to the next slide. And it's always good when you give a talk to acknowledge your funder, but it's not just, that's not the only reason why I put that there. So we applied for competitive research funding, and there's been another call since by the VW Foundation, which was explicitly looking at the role of human environmental interactions. So it was really to understand how humans interact with the environment, especially around infectious diseases. So very much coming out of that, out of experience of COVID, but the VW Foundation has, or this specific call is targeted at transdisciplinary or very interdisciplinary research. So if you're interested in doing that, then the call is they publish calls about once a year. So I recommend you can also look at this. It's a good source for funding. So, but let me just briefly. So I'm the PI of this grant, and formally it is held by my research institution, by my main research home, the Robert Koch Institute. And if you go to the next slide, maybe all of you are familiar with this, but I will just introduce this briefly. Next slide, please. So on my screen, it's still the VW call. Oh, yeah, wonderful. So the RKI is Germany's National Public Health Institute, and it's got a very long history. So one of the things I like to stress is that CDCs or National Public Health Institutes quite often don't have as strong research ethos as the RKI has. The RKI was founded for the then already famous microbiologist Robert Koch. And while it has a lot of government agency and it has a formal mandate, which is enshrined by law in Germany, it also has a very strong research culture and research ethos. So people who work there, like myself, also are research active and apply for these grants. I'm going to try and speak a bit more loudly in terms of the audio. Apologies, I can see the comment there. So next slide then. So the RKI has a long history, and its mandate really is around the protection of health in Germany. But I did want to point out not just GLOHRA is a comparatively recent development, but really it's also in terms of national public health, the realization that that international work is absolutely essential to national public health. So in 2017, following the outbreak of Ebola virus disease in West Africa, the German parliament actually changed the Infection Protection Act, which guides and describes the mandate of the RKI, to explicitly include international work, both bilateral research partnerships, capacity building, and also support to countries around crises. So both research and projects, but also multilateral work and deployment of staff overseas. So this is in a way indicative of the extent to which our international networks and the research that we are doing collectively as a community is essential to national public health everywhere. And the RKI is one of the national public health institutes around the world that has a strong footprint in that. And I'm sort of very happy that we also continue the RKI tradition of doing our more agency-based work, if you want, but also research-based. So the next slide, please. So as a result of that change in the legal framework, in the mandate of the RKI, this center that I now have the pleasure of leading was created, the Center for International Health Protection, and we have a number of different units, 4 units and 2 sort of the German word Stabsstelle. But we cover epidemic intelligence. We have evidence-based public health, which is sort of systematic review function. We are engaged in preparedness and operations support, which is essentially keeping people safe in the field. And we also offer public health laboratory support. We have a strong partnership with the WHO hub, but we're also the national focal point for several of the international activities, such as the emergency medical teams and the GOAN, which is the Global Outbreak Alert Response Network. And we're guided by an independent scientific advisory board. So just to say, global health as a research community is strong, but it's also strong as a community in sort of the public health practitioners. And that is where the SENZOR grant, or where I am based. So next slide, please. So in SENZOR, we, together with colleagues in 22, and very much under the, I would say under the influence, but under the impressions of the COVID-19 pandemic, we articulated a couple of hypotheses. And the first one of that is that climate change-induced changes in the landscape ecology lead to changes in human-animal interaction. So the changes in the landscape and the way in which people use the land and the ecology leads to changes in which animals and humans interact. And that in turn changes completely the types of risk we have for zoonotic spillover. So for new ways in which animals can transmit diseases, or diseases can find their way, viruses and bacteria, from animals to humans. And we, and it's really my sort of very esteemed colleagues who are introduced on this grant, who did much of the sort of preliminary work around that, then thought, okay, to better anticipate these types of risks, we need new ways of understanding and measuring them. And to do that, we need to understand ecology, we need to understand anthropology, so how humans behave with animals. We need to understand the microbiology and the virology that results. And we need to have skills to put all these different factors together into a model that will allow us to both model but also forecast such risk. So this was our hypothesis. And then in a way we articulated the SENZOR project in response to that. I just want to say the people you can see on the pictures, I don't know how large the pictures are on your screen. One is the person at the top is Ali Yu. He's a veterinary PhD in the Gambia, and the middle picture is a colleague from Nigeria working in the lab, and at the bottom is also a picture from a previous One Health project we have undertaken in Nigeria. So just so that you have a that you know where those pictures are from. So in SENZOR, then next slide, please. We wanted to test or sort of work on this hypothesis by generating a new understanding for how zoonotic outbreaks occur, develop this novel approach for assessing risk through rigorous, transferable, interdisciplinary science. So we knew it had to be interdisciplinary, and interdisciplinary science is already one big lesson I want you to take from today. Really can only be embedded in partnership, and often that goes with capacity building activities which work for both sides. So we wanted to have a very strong capacity building element, and you see here on the pictures I have shared with you are of the sites. On the left hand is Nigeria and the 2 sites in Nigeria, and I'll go a bit into why we chose those two sites in each country. And the other is a picture, a satellite image of the Gambia, which is Africa's smallest country around the Gambia River, which is essentially a little line in West Africa is embedded into Senegal along the river. Relatively different. Both countries are in West Africa, but they do have different ecology. But what we wanted to do in both countries was to look at settings, 2 different landscape settings. So we wanted to look at a sort of more, in a way, rural setting, and one where agriculture was more organized, if I can say that. Next slide, please. So formally, the project articulates. Then I think I think if you click further, yes, thank you. So it has 2 aims. One is to create this new understanding, and then also to create an approach to assess risk. And again, so the interdisciplinary science in a comparative approach. So we thought if we develop this approach, if you do it in one country, everybody's going to tell you, oh, it's just one country. You can't transfer it. So we thought, okay, let's use 2 different sites in 2 countries. And it has these different aspects. It has metagenomics to firstly see what kind of pathogens are available in the community, and then focused serology, so focused survey of animal and humans. Once we have identified through the metagenomics specific pathogens we want to focus on. In the same communities, so these 2 different communities in each country, we're going to do anthropological in-depth investigation. And from that, we're gonna then generate a socio-ecological network through which we can then model the pathogens we focus on. And stakeholder engagement is very important because we want to very much, this is all about risk and identifying things that can be used. And then the capacity element that I stressed at the beginning, we decided that we would do the field work in the different sites through a cohort of 4 PhD students. So each of the project partners has a PhD student effectively who's responsible for the primary data collection. And if you go to the next slide, then. So this is the team and the different work packages. The first one you can see is around anthropology. The second is the colleagues in the Gambia, led by Professor Chris Murray. He's an ecologist. And there's a very talented microbiologist, Dr. Abdul Sesai, who leads the genomics platform there. And then in Nigeria, Dr. Anis Happy, who is, again, a virologist who's focused on Lassa fever, actually predominantly. She's also a vet. And she's the PI in Nigeria. And then at the Zoological Society or the Natural History Museum now is Dr. Dave Redding, and he's a modeler. So he's bringing all of this together. So again, maybe the key to say here is there's lead PI in each country in the institution. And as you can see, there are 4 work packages, which are very different disciplines. So we are in different locations, and we all represent different disciplines. And I'll get a bit more to the nitty-gritty of that in a moment. So then next slide. So here you can see basically the work packages again highlighted around the methods we're going to use. So we are now in year 1, and we're doing the baseline. So the baseline involves both understanding the communities from an anthropological perspective. So how do people live in the community with the animals? And it involves pathogen sampling, so the metagenomics. So in the same communities, we look at what animals are present, and we swap. We do wide-scale swabbing, looking for a range of different pathogens. So broadly, before we narrow that down. So and then from the broad swab, from the metagenomics and the anthropology together, we're going to say, okay, we're going to focus on these pathogens and that interaction between human and animals. So it's very much social science and the lab science working together to understand, to see, okay, where are the areas where we want to really go in depth in terms of understanding. And that will then lead our study design in the next 2 years, where we will determine which communities we do the serology around, which will be very focused. So we'll have a smaller cohort, 250 people, around specific pathogens. And we really, then to add to that, we have this landscape ecology. So we want to look at an agricultural gradient. We're looking in each of the countries in essentially in an area, as I mentioned, which is maybe sort of more, they're both quite rural by our standards. I don't want to say one's more rural than the other, but where the use is more industrious of the agriculture than in the other. And then we bring in the next slide, if you see, so we bring this together. This is essentially one slide. I won't go through this again, but this slide brings together the different work packages we have. And we want to understand these different networks, so how people and their animals interact with each other in this environment. And if these networks are different, depending on where you're based, right? So if the network is different, we're going to see is the pathogen that's transmitted different? Are the pathogen networks different? And does that allow us to then forecast where you can say, okay, if the ecology changes in this way, you would see concomitant changes in both the way animals and humans interact and in the way that pathogens spread, right? So that's where the modeling is important to bring this all together. So the next slide maps this out again once more in terms of the structure and the timeline. And here you can see that. So we're in 2024. We've had our preparatory phase. We've done some of the stakeholder engagement, and now we're really the broad social science. So the colleague in Nigeria is in the community now. We have a postdoc there. We're just waiting for the ethical go. The PhD student is already in the Gambia, and we've started with the metagenomics in Nigeria. So this again will then lead through co-design with local stakeholders into the more focused social science and the serology that we'll undertake in 2025 and 26 before we do the modeling, as I mentioned. So this is just to show there's an incredible amount of interaction between these different disciplines. And it's quite a feat to keep that moving in terms of a timeline, right? Because you need the anthropologists to go with the lab scientists at the same time when they enter the community to introduce the project, and the anthropologist remains. But you need to have done the swabbing at that time. Then we'll have to do some preliminary decision and analysis to decide on the priority pathogens and what we're focusing on in terms of where we're looking at that connection. Okay, so I think, or I hope I've given you the impression of that. Next two slides are just a little bit more in depth on the anthropology. So if you go to if you click through, so there's an animation in there, water. Yeah, so there again, what we really want to understand is this interdependence on the landscape that is changing. And we want to know about how people behave with their animal and whether that contact changes and what even is a contact. So for example, I'll try and give you a very simple example. So if it's starting to become hotter, do people sit perhaps underneath a tree more in the shade? And then if you have bats in that tree, the dropping from the tree will pose an increased risk for the kids who sit there more at lunchtime. So this is a very, very simplified understanding of that. And we do this very in-depth ethnography for that, and we'll do social network analysis really of animals and humans. And for me, that's exciting. As a social scientist, that's quite an exciting thing, because normally social network analysis has focused a lot on understanding human-to-human networks. But here we try to identify human-animal interaction and how that changes based on the differing ecology. So just to bring this to life a little bit more, I hope it wasn't too abstract now, is next slide. In the Gambia, there's comparatively little research data on zoonosis and zoonotic risk. And what we decided to do, and we did this during the initial phase of research after understanding the context better, is that rather saying, okay, rural, urban or something like that, because the river has such a central role in terms of how landscape is used, the interaction with the landscape, we chose one community which is closer to the river and one which is further away from the river. And then we're going to look at the changes in land use, in livelihood activities, in water availability. And one important thing, so one additional complexity I had failed to mention is because we're looking at ecology, we're trying to capture different seasons in each of the sites. So the anthropology has to happen over different seasons to see how contact, again, is varying by seasonality. That's very important for the ecological gradient. And then the next slide just gives you satellite images of the study sites around the river Gambia. That's the more natural side closer to the river, and the next slide is the further away. And I think maybe you can see immediately the already differences in the way that those sites present in terms of the farming activities that we could map even before we begin the baseline. So we wanted to do something very ambitious, right? So this is hypothesis-driven research where we really wanted to develop a novel understanding. And you can see the complexity we have there of the different disciplines to really understand how you can see where risk might emerge, right? So how can we understand where new risk might emerge? And I can't present the findings yet, so I'm sure we won't quite get as far as we intended to. But I think already understanding, putting the pieces together, the ecology, the human and the animal interaction, and how this changes and how it changes risks, we hope we will make a significant scientific contribution. So I'm my summary slide, and what I wanted to leave you with sort of in terms of reflections for the discussion is again, for this grant, it's very obvious for anyone living in communities, but I think everywhere in the world now, that changes in the natural environment mean we see changes in human and in animal behavior. And we also see changes in how humans and animals interact, right? So if we want to put a One Health approach into practice, we have to really holistically look at that. And what this means is it means on the one hand, we'll see different and new risks emerging, which is important if you are a CDC and your task is to think about risk and assess that and try to mitigate it. But we do not yet have strong frameworks or conceptualizations around how do these factors interact. So how do these changes affect one another? And then to have this kind of conceptualization is really the beginning to get to understanding risk. But to generate this understanding, and that's really the key point of this project, and the key point of I guess my talk today is you need interdisciplinarity, right? So you cannot do, even if you're the cleverest and smartest virologist, you know, who knows the community well. You cannot gain this understanding if you do it on your own. And if you can make important scientific contributions if you're a social scientist and you do this network understanding. But really to understand and generate a framing around this is you do need the interdisciplinarity. So maybe in the discussion we'll get into this. You can imagine that a lot, I think I've shown you, there are a lot of moving parts in this grant, and they have to align, right? So the communities have to align, the sampling has to align, the seasonality has to fit. So it's hard to get all of those pieces moving in the right order. And that's, I hope the other thing we'll get to is really also it's fun. I mean, it's very rewarding, the exchange with the other disciplines. Yeah, and that's it from my side so far. Thank you.